Okay, so I want to talk about what the macula densa is. The macula densa cells are actually part of what you call the juxtaglomerular apparatus. And what is the function of this macula densa cells? Well, if you look carefully, the macula densa cells are actually part of the distal convoluted tubule. So let's look at the nephron again. Remember, the nephron has the PCT, which is the proximal convoluted tubule, right? Eventually, we're going to reach the loop of Henle. This is the descending loop of Henle, right over here. Then we're going to hit the ascending loop of Henle, right? We're still in the water slide. And eventually, we're going to hit the distal convoluted tubule. And the first early part of the DCT, we actually call this the macula right over here. Let me just maybe put a different color. I'm going to put a, actually I'm going to put a dark green. This part over here, which is the early part of the DCT, it's called the macula densa. Actually, not this part. I'm just going to write this really small. So what is the purpose of the macula densa? And just to let you know, in reality, this macula densa actually wraps all the way around, and it actually is somewhere around here. So this macula densa, it's drawn over here so you can see it a lot easier, but in reality, it sits right by the Bowman's capsule. So in reality, you see this DCT portion? It actually sits kind of around the Bowman's capsule. This whole thing, this is essentially the Bowman's capsule right over here, right? This is the Bowman's capsule. All right. So what is actually happening here? Well, there's actually these pressure sensors right on the DCT. And when the pressure is way too high, let's say there's a lot of blood flow in this region, right? I'm sorry, not a lot of blood flow, a lot of filtrate flow in this region. Let's say there's a lot of fluid flowing in this region, right? And there's a lot of pressure on these walls because of all that fluid flowing inside the nephron. Well, do we actually want more fluid to go in here or less fluid? Right? So think about this. Think about you have a balloon, right? And there's a lot of pressure on the balloon walls already. Do you want more air to go into that balloon or less air? Well, you don't want it to pop, right? So you're going to actually have less air go into that balloon. In this case, we actually want, if the pressure's really high, and that's detected by these macula densa cells right over here, it's going to say, hey, we don't want extra filtrate to go right through this distal convoluted tubule. So we're going to decrease our GFR. How are we going to decrease our GFR? Well, we said before that one way to actually decrease GFR is to actually vasoconstrict our afferent arterioles, and that's what actually happens when pressure is really, really high in this nephron, as detected by the macula densa cells. This actually gets constricted, and less fluid goes right into the kidneys. In other words, GFR actually decreases.